This protocol enables the rapid assessment of NF-kB and AP-1 activity in reporter macrophages cultured on adsorbed protein layers, and the contribution of cell signaling pathways like toll-like receptors to that response. The main advantage of this technique is its simplicity, allowing cell culture supernatants to be quickly analyzed for NF-kB and AP-1 activity using a straightforward enzymatic assay.
Begin by dissolving PMMA in chloroform at a 20 mg per milliliter final concentration in a 20 ml glass scintillation vial in a fume hood. Place a magnetic stir bar in the vial and stir the mixture for at least 2 hours. When all of the solids have been dissolved, transfer 400 microliters of the PMMA solution onto the center of each borosilicate glass microscope slide per plant condition on a spin coater, and spin the PMMA onto the slide for 2 minutes at 3,000 rotations per minute. Then store the spin-coated slides in a 70% ethanol-sprayed clean box. Next, in a biological safety cabinet, use sterile forceps and aseptic technique to attach 8-chamber sticky wells to the PMMA-coated slides, pressing firmly on the top of each sticky well to make sure they are strongly attached.
Take care to line up the sticky wells with the edges of the microscope slide so that all of the wells attach to the slide and don't leak.
Incubate the sticky well-attached slides at 37 degrees Celsius overnight to secure the seals. Then add 200 microliters of cell culture-grade endotoxin-free water to each well for a 60-minute incubation at room temperature to test the seals the next morning. At the end of the incubation, aspirate the water, taking care not to disturb the PMMA coating. Wash each well 3